So here in the Netherlands, the LSAMP students have their weekly meeting with one of us cooking dinner. So today, Miguel is cooking what?
I'm cooking steak, chicken, rice, some vegetables.
Okay, cool. Okay, so this is the beautiful plate that Miguel made, and here's everybody else's plate.
Oh, okay.
And so we'll get people's reaction. Okay, so tell me an update this week about lab.
Okay, this week is an exciting week in the lab, and the goal for the week is to transfect some of the cardiomyocyte cells that we have with foreign DNA. So that's really cool. That's really exciting. Also, we will attempt translocation of receptors inside of a cell. So we're really gonna, for lack of better words, jack up an existing cell. And so that is exciting. And then, we're gonna, based on how we're gonna modify these cells, we're gonna try different inhibitors and see and kind of play with that a bit. So that's exciting, to study how these modifications affect disease states like obesity and type 2 diabetes and insulin resistance.
Right, Miguel, so tell me about, well, for the past 2 weeks, what did you do?
For the past 2 weeks, so the beginning I was reading a lot of articles on my project, which is, it has to do with compounds or cytokines responsible for inflammation. And so it's actually a maladaptation in dyskinesia. And so we're looking at the actual pathway of these cytokines and actually the pathway to inhibit the maladaptation, which is P38 MAP kinase. And so my job is to look at interleukin 6 beta, which is one of the cytokines. And I'm looking at, right now, since we don't have our rats, and we can't, we had a little snag that the rats were supposed to come in like a week ago, but now it's coming in 3 weeks later. So until the rats come in, I'm testing a lot of the tissues that he already had from human patients, and I'm testing out antibodies antibodies for interleukin 6 that should be present in those tissues, or interleukin 6. I just got confused. Edit that part out. I'm sorry. Anyways, we're testing the antibodies with these tissues to see if interleukin 6 beta is present and if the antibody works. And then after that, we're going to purchase the antibody from Santa Cruz, and then test probably some more human tissue. But then when the mice actually come in, that's when we'll really start what we really want to do, which is induce this inflammation by nipping the spinal cord, and it'll create this inflammation in the mouse. And so we're going to see what cytokines are activated in what parts of tissue around the spine.
Samsia has only been in the lab for 1 day, so just give us a short update what you did today. You did a lot today for your first day.
Today was my first day in the lab. At the beginning I was getting a little bored, but seems like there's going to be a lot of work. So in about 4 or 5 hours, it seems like we've done a whole lot. I have 3 publications and 1 paper, plus 1 non-published paper to read for tomorrow. And I already went through the technique with one of the undergrad. And tomorrow I'm going to go over one of the MDs, and the following that I'll be on my own.
Oh, what technique are you going to do?
Initially I thought we were going to work on bladder pressure and stuff like that, but this had nothing to do with the bladder pressure. This is working with antibodies to be able to find a certain prostaglandin inhibitor called the 5I and 2I. He already worked with the 2I, but we'll be working on the 5I. So initially they thought that type of prostaglandin was found in the muscle around the bladder, but they found that it's not actually in the muscle around the bladder, but it's actually in the bladder cells itself. So they're doing some type of stains and immunochemistry to be able to to prove that it's actually in the cell of the bladder, not in the muscle around the bladder. So that's basically what we're doing: staining and measuring, making solutions, titration, a lot of stuff. But I think it should be fun. It sounds it's gonna be a lot to do, but it sounds doable.
Okay, Henry, let me know, how was your first 3-4 days?
Week.
4 days.
It's been about a week now.
Well, actually, very interestingly, once I got here, Dr. Steinbush decided to place me into the lab, which I thought it was a great idea, just because he actually took a minute to talk to me and ask me about my interest, and after talking, we decided on a lab that's actually doing work on the same protein that I'm working back in New York. Excuse me. And very interestingly, I actually started working with one of the doctor students here now. His name is Fabian, and we're working on BDNF, brain-derived neurotrophic factor, and how the pathway that it undergoes via the TrkB receptor. So we actually want to see what's the downstream cascade after that, after it binds. And we also want to try different antagonists and agonists and see whether we could replicate the signaling pathways down the road. As of now, I helped in a few surgeries of animal sacrifices where we removed the brain, and the hippocampus was removed and stored, as well as the frontal lobe, but that was for another project that they're actually working on the side. I'm very interested about learning the technique of Western blotting, which is one of the things that I want to implement into my own research. And I will actually begin tomorrow to prepare the samples, doing a Bradford's assay, which I've never done before, so it will be really interesting to start it because it's labeling the proteins and quantifying it. So that's the first step of Western blotting. Then after that, Thursday and Friday, I'll actually watch the first complete Western blot be run, and next Monday I should be starting my own.
Okay, so I guess I've been doing like jack of all trades in the lab. I work with Leonidas, and we do DNA methylation on epigenetics and Alzheimer's disease. But the thing is, last week he was on vacation. So the first week I helped him with DNA methylation because we wanted to know if in the brain, like different proteins are different in Alzheimer's disease patients versus normals, and we also use rat models. But then he went on vacation, so last week I actually followed Fabian, so me and Henry did almost the same thing. He was my PhD student first, but I helped him the first couple days with chromatin immunoprecipitation. That was interesting because we had to actually, he was growing it, and I got to actually scrape the actual cells off the growing plate and see how much was floating inside, so that was cool. And then at the end of the week, we dissected brains. We did 148 brains in 3 days. So this week, Leonidas is back, so I hope to do a DNA methylation of my own and use that hopefully for what I do for my PhD back home, which is in obesity. So hopefully we can incorporate epigenetics into obesity, what we do with conditioned taste preference.
All right.